So I'd like to introduce our company, Cartherics. It's a small startup that we've founded in Melbourne, and our mission is quite modest. We think where we're heading is the future of cancer immunotherapy, cancer treatment. And we were founded in 2015. We have our laboratories at Monash. If you've been to Melbourne, Monash University has a purpose-built facility called the Monash Health Translation Precinct. It's a 7-storey building with commercial and academic research combined with high technology platforms, and a floor where we can do our clinical trial, a clinical translation. So it's a purpose-built research to the clinic building, which is actually fantastic. We've raised over $13 million now, and combined with Series A and Series B and government grants. And we began our operations in the lab in January 2016, which doesn't seem that long ago, but it's been very active time since then. So our core technologies are basically threefold. We have our basic technologies in T-cell biology, understanding how the thymus works, how T-cells work. But a very big, strong part of our work is led by Alan Trounson, our CEO. Many of you know Alan as the president of the California Institute of Regenerative Medicine from 2007 to 2014, and his work in IVF and embryonic stem cells. So we have that mix between immunology and stem cells straight up. But Peter Hudson, if I can get this laser to work, Peter Hudson is a world-renowned protein biochemist, particularly strongly in antibody structures, and he is a phenomenal support for us in our designing our scFvs, tweaking them to be high affinity and low immunogenicity. Ian Nisbet is a corporate advisor. He's had 25 years and more of experience in this field, with a lot of immunology as well. So we call him the Minister for Keeping Secrets, and he guides us through all our dealings. Maureen Howard has a strong history in clinical trial management programs at Stanford, particularly involved in the CD47 program. Miles Prince is a leading oncologist, hematologist from Melbourne. Many of you will know Miles. He's a global leader, particularly in T-cell lymphoma. And Lee Dayton is a leading Australian journalist who deals with our comms for us. So in the present process of preparing for 2 phase 1 trials, phase 1, phase 2 trials. The first is in cutaneous T-cell lymphoma, and that is our CAR T-cell is an anti-TAG72. I'll tell you about that a bit more in a minute. Our second clinical trial is in relapsed ovarian cancer, also with the key target of TAG72. So TAG72 has been around for quite a while, but sort of a sleeper. It stands for tumour-associated glycoprotein 72. It's an oncofetal antigen. It's present very broadly throughout the embryogenesis fetal life, but disappears in adult life. It's basically negative in adult tissues, but it's a very strong staining and characteristic of adenocarcinomas. And we've also found recently that it's present on T-cell lymphomas, and this is the brown staining showing the strong staining of TAG72 in a tumour biopsy. So how do our CAR T-cells work? Well, we prepared them, we used a number of models, this is a Nodskid gamma mouse model transplanted with a human ovarian cancer cell line of CAR3. And we allowed the tumours to take, and when they get to about 100 millimetre cube size, subcutaneous, we infuse 3 injections of the CAR T-cells. And you can see on this here is non-transduced control T-cells. When you give the T-cells, that slows down the tumour a little bit, but then it takes off, and basically animals have to be culled very quickly. When we add our CAR T-cells, the TAG72 CARs, there's an immediate cessation of growth of the tumour. When you normalise those data, it's basically up to about 60 days, there's really no cancer left. And that's pretty impressive for a solid tumour, bearing in mind we're going after solid tumours, but we wanted to tweak the system a little bit more because we realise, in fact, that there are 3 important parts of CAR T therapies. First, you have to have a good target, and we think TAG72 is a good target. The second is you have to supercharge your CAR T cells so that they can better attack solid tumours. And what we've done in that regard is we have a number of novel genes that we've knocked out and examined what effect they may have on the CAR T function. And we focused on immune suppressive genes. This is one such example. So again, we've got our TAG72, our non-transduced T cells. This is the same animal model with the Cartherics growing. And what we've done in this system is actually to reveal the enhancement effect of the knockout gene. We allow the tumours to grow to a larger size initially, now around 200 millimetre cubed, and we give about half the dose of CAR T cells. So in this model, the non-transduced controlled T cells, the cancers take off and the animals have to be culled. With the TAG72 CARs, now under this more stringent conditions where they have to attack a bigger cancer with a lower load, the cancers start to relapse. But when we add the gene knockout to these CAR T cells, we can completely ablate that cancer. And in fact, it's almost impossible to find it. If we go back to this model now, we take the animals which were relapsing and we give them back fresh CAR T cells. There's an immediate drop in the tumour growth, but again it relapses. But if we give the combination of the TAG72 CAR with the knockout gene in it, again these cancers relapse, come back down to almost baseline. We've shown this now in multiple donors, and really you basically can't find anything in the tumour mass at the end, and this just shows you that. This is the TAG72 knockout CAR T cells. In the site of the tumour, these are the T cells, remnant T cells. There's virtually no human, this is mouse tissue, there's virtually no human tissue left at all. In the control non-transduced T cells, it doesn't show up very well in this slide, unfortunately, but there is still quite a lot of TAG72 positive cells. So in summary, the combination of the powerful anti-TAG72 combined with the gene knockout has really supercharged these T cells. They not only kill the cancers to the point we can't find them anymore, but the T cells have a much longer longevity. And I think that's a very important key to our work coming up in solid tumour models. So we also quite fortuitously realised that TAG72 was expressed on T-cell lymphoma, and a significant portion of, about up to 40% of T-cell lymphoma patients show elevated levels of TAG72. It was very surprising, given its restriction up to that point in time on adenocarcinomas. And the cutaneous T-cell lymphoma patients have very limited therapeutic options, and so we are planning a clinical trial on these patients. And this just shows the distribution of TAG72. These are circulating T cells. This is the leukemic form of cutaneous T-cell lymphoma. And you can see there's quite a high level of expression of TAG72 in the patients compared to healthy donors. And the restriction of the expression is to CD4 positive cells, which aligns with the predicted leukemic form of the disease. These CD4 positive T cells are proposed to enter the skin and cause the lymphomatous reaction. And we can take our TAG72 CAR T cells and kill these TAG72 bearing peripheral blood T cells. And this is a control showing the same killing of a Jurkat cell line, which is also strongly TAG72 positive. So where we're heading with this actually is to develop an off-the-shelf allogeneic product, like I think many in the field are now heading towards. We start with homozygous haplotype, HLA haplotype cord blood cells. So these are these triple homozygous so-called super donors from which we create iPS cells. We then can introduce the CAR T cell, the CAR construct. We can modify, we can sculpture out the genes we don't want. We can then clone these iPS cells and store them as a master bank, or directly differentiation into the therapeutic product as an off the platform target. So we're preparing a first-in-human clinical trial at the moment using allogeneic CAR NK cells, in fact, directed from iPS cells, and I'll show you that in a minute. And this program is being developed through a government grant that we got for around $3.3 million, and with a number of partners in Melbourne, particularly Cell Therapies, who will be in charge of the method of cellular manufacture. So how do we make our iPS to NK cells? We start with the iPS cells, we expand them, we pattern them towards hemogenic endothelial development, and then from that point in time we could actually direct their differentiation either into alpha-beta T cells or into NK cells using different combinations of media and combinations of growth and cytokine factors. And from the NK progenitors we then change the medium into an NK expansion. And this whole process takes us about 30 days. It relies on very important patterning at the beginning to mimic the NK status development that we see in the body. And of course our focus is on a xeno-free, scalable, molecular defined and clinically translatable system. And from every point from here to here, that's true. And at the moment we can get about 150,000 of these iNK cells per individual iPS cell, which from a point of view of scale-up becomes quite feasible. And just to show you how they work, so this is the Excelligence in vitro cytotoxicity assay. These are of Cartherics, the TAG-CD2 high expressing cells growing just in medium alone. But if we add NK cells either from cord, from adult or from iPS-induced NK cells, you can see there's quite a strong killing of these cells. And if you look at mesols, which are also an ovarian cell line but have low levels of TAG-72, the CAR T cells will kill these cells, but they are ineffective against these mesols. But when we add the NK cells, we get strong killing of both the low and high expressing TAG-72 cell line. So then we thought, well, we're already getting very strong killing with the NK cells by themselves, induced from iPS cells. So we thought we'll challenge the system and use a lower effective to target ratio of these NK cells and see whether we can reveal an additive advantage of the CAR. And when we do that, so these are the cells growing alone, the mesols growing alone, the iNK cells at a much lower effective to target ratio, we can show that they are in fact slowing down or even killing the cells. But when we add the TAG-72 CAR onto these iNK cells, we get almost complete ablation. So that's pretty much our proof of concept of combining the technologies of iPS to NK cells and the TAG-72 CAR. And in addition, we're knocking out this immune suppressor gene to make these even more powerful. And we think that that's the sort of combination that you're going to need to have an effective cell-based therapy for dealing with solid tumours in vivo. And our model, of course, is ovarian cancer. So just in summary then, that was a very quick summary, here we are back in. So we're very fortunate to have some very strong partnerships. Cell Therapies, I mentioned, a collaborative research centre partnership. They'll be in charge of manufacturing our CAR T cells. We have a licensing agreement with Tulgen, which is a Korean company. We have, through them, partnership for gene editing. Pancella is a Toronto-based company which has technology for immune cloaking that is preventing rejection of these allogeneic cells, combined with the fail-safe technology, almost a suicide-type switch. And Berry Genomics in Beijing is our partner for our genomics. And I must say, from the extensive number of meetings we've had this week, like I guess all of you, this speed dating, we hope to be adding a few more partners under this in the coming weeks or months. And I'll leave it there. Thank you very much.